Viruses are terrifyingly efficient. Inside a host, a single viral invader can hijack a cell's machinery and violently churn out thousands of copies in hours. For a long time, biological dogma told a very specific story about how we survive. Because human beings reproduce slowly, taking decades to pass on our genes, scientists assumed we relied on a complex, uniquely adaptable immune system to keep up. We have a first line of defense known as the innate immune system. It acts like a biological tripwire, a fast-acting network that immediately detects foreign pathogens and triggers an inflammatory response. Because these immune proteins are so sophisticated, researchers widely believed they were recent evolutionary inventions, exclusive to complex multicellular life forms like us. Then, in 2013, biochemist Zhichan Chen mapped a crucial pathway that serves as the central alarm system for human cells. When a virus invades, it leaves foreign liaison floating in the cell. cGAS detects and locks on to that misplaced material. Once triggered, cGAS synthesizes a secondary signal called cGAMP. Those particles activate STING, setting off a cascading inflammatory response. It was a brilliant and highly specific biological mechanism, but it also created an evolutionary puzzle. Science had no explanation for how such a remarkably complex pathway actually came to be. Microbiologist Philip Kranzush wanted to find out where this alarm system came from. He started looking for other organisms that produced that secondary signal, cGAMP, and he found it in a totally unexpected place: simple single-celled bacteria. At first glance, the genetic code for the bacterial enzyme and the human enzyme looked nothing alike. Out of a 300 amino acid long sequence, the bacterial and human versions shared a tiny overlap of only 5 or 6 amino acids. This diagram compares immune protein shapes across species. The human STING protein's core matches the ancient bacterial version. Over billions of years, the protein's outer edges, the dashed outlines, mutated to evade viruses, but the core physical shape remained unchanged, allowing the alarm to work. This structural match revealed that the core of our innate immune system was developed billions of years before the first humans. Our frontline defense relies on ancient bacterial technology operating inside our own cells. Around the same time, microbial immunologist Wormtim Sorek developed a computational pipeline to scan bacterial DNA. He discovered massive defense islands, dense clusters of immune genes hidden deep inside their genetic code. These islands were forged in a fierce billion-year biological war between bacteria and bacteriophages, the viruses that hunt them. With trillions of casualties every day, the pace of evolution in this microbial battlefield is incredibly fast and utterly ruthless. Many of the defense mechanisms they developed are strictly suicidal. When the bacterial cell senses a viral invasion, it triggers a system like cGAS-STING to intentionally destroy itself, preventing the virus from replicating and spreading to the rest of the colony. Viruses evolved a countermove, deploying specialized molecules to soak up the bacteria's cGAMP alarm signals. In response, bacteria evolved the panoptes mechanism, flooding the environment with decoy signals. The viral sponges absorb the fakes, allowing the true alarm to trigger self-destruction. This relentless, high-speed evolutionary cauldron forced both sides to innovate constantly. That exact trial by fire is what shaped the most crucial weapons operating in human biology today. So how did we get them? Complex eukaryotes, like humans, didn't invent these immune tools. We acquired them over 2 billion years ago, when an early ancestral cell engulfed an ancient bacterium, which eventually settled in as a mitochondrion and seeded our entire genetic lineage. Bacteria function as a massive evolutionary makerspace, a cauldron where new molecular weapons are forged and constantly refined for all of terrestrial biology, and eukaryotes are still borrowing their technology. Take the Drosophila fruit fly. Several million years ago, it horizontally acquired a toxin gene originally used by viruses. Today, the fly uses that stolen toxin to poison parasitic wasps that lay eggs inside its larvae. For complex, slow-breeding organisms, borrowing tools from rapidly evolving microbes is an essential biological life hack. It allows us to cheat evolutionary time and stay ahead of our own microscopic enemies. Today, researchers are using those ancient bacterial defense islands as a predictive roadmap. Because experimenting on simple bacteria is highly efficient, Scientists can test hundreds of new antiviral systems in petri dishes, and then use computational models to pinpoint entirely unknown immune mechanisms still hiding inside human cells. The rules of engagement for our survival were written billions of years ago in microscopic warfare. Tracing this ancient microbial history is now providing the blueprints for the future of human medicine.